Hello and welcome. Now we will discuss some key bioinformatics concepts such as reads, single-end and paired-end sequencing reads. Let's start with a basic concept. What is a read? A read is the actual sequence of nucleotide bases A, T, G and C generated by a sequencing instrument from a DNA fragment. In simple terms, when a DNA fragment is sequenced, the machine records the order of bases and that recorded sequence is called a read. A read is the fundamental unit of sequencing data. A read is not just a biological concept, it is also digital data. Each read is stored as text inside a file, and these files are used for downstream analysis such as alignment, assembly, and variant detection. In Illumina sequencing, each cluster on the flow cell corresponds to one DNA fragment, and a single read is generated from each cluster in single-end sequencing. Sequencing happens cycle by cycle, and each cycle determines one base in the read. During sequencing, that cluster emits signals. As cycle proceeds, these signals are converted into base calls. So, a read is built base by base as shown here. Now, let's look at an example. Imagine the sequencer running through cycles, and in each cycle, one nucleotide is identified. Suppose the sequencer outputs this after the completion of all cycles, then this sequence is a read. Each read represents a portion of the original DNA fragment, and millions of such reads are generated in parallel. Because genomes are very large, millions to billions of reads are required to reconstruct or analyze the full sequence. Importantly, a read is not just a sequence of bases. Each base in a read is associated with a quality score. This score reflects the confidence of the sequencing machine in calling that base. We will discuss this later. So, sequencing produces both sequence and quality information. Now, what is read length? Read length is the number of nucleotide bases determined or sequenced in a single read. Simply, read length refers to the number of bases in a read. If we take the same read as before, let's count the bases. There are total 32 bases. So the read length is 32 base pairs. Now let's connect this to Illumina sequencing chemistry. In sequencing by synthesis, each cycle adds one nucleotide and the total number of cycles determines the read length, as we can see here. So 50 cycles produce a 50 base read, while 150 cycles produce a 150 base read. That means the number of cycles determines the read length in Illumina sequencing. Now let's connect this output back to sequencing strategy. These reads can be generated in two ways, single-end sequencing and paired-end sequencing. But what exactly does that mean, and why does it matter for analysis? Let's start with single-end sequencing. In single-end sequencing, each DNA fragment is sequenced from only one end. This is shown by this single red arrow here. So for each DNA fragment, we get just one read, that is one read per fragment. For example, 150 base pair read as shown here. This is simple and efficient, but it gives us limited information. Now look at the paired end reads just beside it. In paired end sequencing, the same DNA fragment is read or sequenced from both ends, producing two separate reads. Here the same DNA fragment is read from both ends, as we can see here. This is read 1, this is read 2. First the read 1 is generated from one side, then the read 2 is generated from the opposite side. Notice how the arrows face toward each other, as we can see here. This means both reads are coming from the same fragment but from opposite direction. This provides two complementary reads for the same DNA fragment. For example, read 1 is 150 base pairs here, and the read 2 is also 150 base pairs here. This is written as like this, that is 2 into 150 base pairs. And this extra information is powerful. It improves alignment accuracy, helps resolve repetitive regions, and increases confidence in genome mapping. Now let's move to the right side of the slide. In single-end sequencing, each fragment produces only one short read, as shown by this colored segment here. During alignment, these reads are independent, as we can see here, and we don't know what lies beyond each read. But in paired end sequencing, each DNA fragment produces two reads read 1 and read 2 as shown by this colored segment here. For fragment 1, this is read 1 and this is read 2. For fragment 2, this is read 1 and this is read 2. And for fragment 3, this is read 1 and this is read 2. If the genome has repetitive regions, it can be difficult to place reads accurately, but in paired end sequencing, we have two reads from the same fragment as we can see here with a known approximate distance between them. For example, notice this central region here. This represents the unknown sequence between the two reads. During alignment, even if one read maps ambiguously, the second read helps confirm the correct position. So paired end sequencing not only improves alignment accuracy, but also helps us infer structural information about the DNA. So to summarize, single end sequencing gives one read per fragment, simpler but less informative. On the other hand, paired end sequencing gives us two read per fragment, more complex but much more powerful. And this is why paired end sequencing is widely used in modern genomics and bioinformatics analysis. Now let's go one step deeper. So far we have seen that paired end sequencing generates two reads, but what happens when these reads actually overlap? Now we are going to understand overlapping paired end reads in sequencing. Let's start with the DNA fragment. In this diagram, you can see a double-stranded DNA with adapters on both ends as we can see here, and a target region in the middle. Now in paired end sequencing, we generate two reads, that is read 1 or R1 and read 2 or R2. The read 1 starts from the forward primer on the left side. It reads in the 5' to 3' direction, moving towards the center. So we get read 1 as we can see here. On the other hand, read 2 starts from the reverse primer on the right side. It also reads in the 5' to 3' direction, but on the opposite strand. As a result, we get read 2 as we can see here. So effectively, read 1 and read 2 are moving toward each other. Now here is the important part. If the DNA fragment is short enough, the two reads will overlap in the middle. As we can see here, this overlapping region is shown here. Both read 1 and read 2 are reading the same sequence in this region, but there is a catch. Read 2 is in the reverse orientation. So before we compare them, that is read 1 and read 2, we need to reverse complement the read 2 sequence. In practice, different downstream bioinformatics tools perform this step automatically. Once this is done, read 1 and read 2 are now in the same direction as we can see here. Now we align them and check if the bases match in the overlap region. Again, this alignment and comparison step is automatically handled by downstream bioinformatics software. If they match well, we can confidently merge or stitch these reads together. This gives us a longer and more accurate sequence like this as we can see here. So to summarize, read 1 reads from one end and read 2 reads from the other end. They overlap in the middle. We reverse complement read 2, then align and merge them, and all of these steps are handled automatically by downstream bioinformatics tools, and that is how overlapping paired end reads improve sequencing accuracy. Now let's look at a slightly different scenario. What if the DNA fragment is longer and the two reads don't overlap? In this case, read 1 and read 2 are still generated from the opposite ends of the same fragment. Read 1 reads from the forward primer in the 5' to 3' direction on the top strand, and read 2 reads from the reverse primer in the 5' to 3' direction on the bottom strand. So again, the reads are moving towards each other as we can see here or here, but this time they don't meet in the middle as we can see here. Instead, there is a gap between them. This is the unsequenced region as we can see here or here. Now just like before, read 2 is in the reverse orientation as we can see here. So to compare read 1 and read 2, we reverse complement the read 2 to bring it into the same orientation as read 1 or R1. Once aligned, we can map both reads to the reference genome. Even though the middle region is missing, we still know that these two reads are connected. As we can see here, final sequences are separated as non-overlapping reads, indicating unsequenced region in the middle. And importantly, we also know the approximate distance between them. This distance information is extremely useful. It helps in detecting insertions and deletions, identifying structural variations, and improving genome assembly. So in this case, the reads are not merged. Instead, they remain as two separate reads with a known gap in between. And together, they still provide valuable information about the original DNA fragment. Now that we have understood how sequencing works and how reads are generated and analyzed, so we have learned about the key bioinformatics concepts behind sequencing data. We started with what a read is. A read is the sequence of nucleotide bases generated from a DNA fragment, and it represents the fundamental unit of sequencing data. Then we explored read length, which is simply the number of bases in a read, and we saw that it is determined by the number of sequencing cycles. Next, we connected these two sequencing strategies. In single-end sequencing, each fragment produces one read, simple but limited in information. In paired-end sequencing, each fragment produces two reads from opposite ends, providing more context and improving analysis accuracy. We then went deeper into paired-end reads. When the reads overlap, they can be merged to form a longer and more accurate sequence. And when they don't overlap, they still provide valuable information through the known distance between them. So overall, sequencing generates reads. Read length defines how much sequence we obtain, and sequencing strategy determines how much information we can extract. And together, these concepts form the foundation for downstream bioinformatics analysis. So that's all for now. Thank you everyone.